Note that despite all these things, by using various methods of aseptic techniques, contaminations are still possible. Okay. And the three methods that I have already described, looking at the culture media for turbidity, looking at the color of the media, and looking for microscopic changes, morphology. These are golden things that one has to always do every day, check every day, in order to catch any early contamination or to be aware that the cells are perfectly healthy. One thing I have to tell you that yellowness of the media does not always mean that there is contamination. If you fail to passage cells in time, if the cells become overconfident, then also they exhibit yellowness. Okay, so if there is any suspected cloudiness within the media, for example, in your original media bottle, then one way to check it is you plate a few ml of that media in a separate plate, separate dish without any cells, and then you leave it in the incubator for 24 hours or 48 hours. If that media changes color or it shows increased turbidity, then that means your media bottle is also contaminated. Okay. None of the aseptic methods that we have discussed, none of them are suitable for the detection of contamination by mycoplasma. Okay, so these mycoplasma contamination are very difficult to detect. They do not change the pH of the media, they do not cause turbidity, the media remains yellow and red in color. So, and they do not form colonies also. So even if you microscopically look, you may not find colonies like a fungal colony that you can not typically see. And the worst thing is mycoplasma contamination. Once it happens, it can spread very rapidly from one culture to another culture. It takes no time. In fact, from one lab, if all the labs are sharing, if in one incubator, all the remaining incubators within 1 to 2 days, they will be completely contaminated. Now, what are these mycoplasmas? Some biological features. They lack rigid cell walls. They can have a generation time of 1 to 9 hours. They are very slow growing. Okay. But then they are very fast as far as spreading is concerned. So they have a very long lag phase, that is true. But then so when you want to detect them and you need to keep the agar plate on which you can detect whether mycoplasma contamination is happening or not for more than 1 week. Okay. Mycoplasma are the smallest self-replicating bacteria. They are less than 0.8 micron in size and they can be filterable through 0.45 micron filter. So that's why I said if you want to do media sterilization by filtration, the pore size should be less than 0.2 microns. The most common of the mycoplasma that one sees in labs and all is a Mycoplasma aureale. Okay, and this is generally coming from the oral cavity of humans only. So one can say humans themselves are the major source. So if you are chatting too long and in fact when you're chatting and spitting in the cell culture room, then essentially you are releasing this Mycoplasma aureale into the environment, which can easily contaminate your cultures. Okay, in fact, 20 to 40% of Mycoplasma infections, not just anywhere throughout the world, that comes from this Mycoplasma aureale. The other group of common Mycoplasma contaminants is the bovine group of Mycoplasma. So this probably comes from the FBS that one purchases, Mycoplasma arginini and Echoleplasma laidlawi. These are mycoplasmas which generally are bovine mycoplasmas which may have not been filtered out during the process of manufacturing and packaging of this FBS. All the FBS that we purchase, if you look at the bottle, it is clearly written that it is filter sterilized using 0.2 micron filters. But still, if there is any poor quality control, then you can have these mycoplasma coming from the serum. It's very difficult to get rid of these mycoplasmas once they are infected, once your lab is infected. And one cannot detect them also. So if we have mycoplasma contamination and we continue to do our experiments, then we don't even know whether our results are actually accurate or not. And many of the results that you had early on established may no longer be working. Or maybe earlier you had maybe for something at 1 micromolar working perfectly fine, and you know that 1 micromolar is inducing your protein by tenfold or causing cell death or whatever experiments you are doing. Maybe early on 1 micromolar is working, but nowadays now you are seeing after 6 months you have established everything, and now after 6 months you are seeing that 1 micromolar is no longer working. You have to increase it to 10 micromolar, and then you are finding the result or something like that. So which tells us that something happened. What has been established at a particular concentration, what you know are having, or for example detection of a certain protein. You stimulate the cells, and after 4 hours or maybe after 8 hours that RNA levels are increasing, and you know that after 24 hours the protein is increasing, you know that. Nowadays you are seeing, earlier and 24 hours you found a 4-fold increase in the protein. Nowadays you are only seeing 2-fold increase, or the time you are seeing you have to incubate it longer to see the same effect. Okay, so that means sometimes we ignore these things and we now change the conditions to the new conditions and then continue doing experiments. So this is very wrong in science. Okay, so one has to understand why what you have been established earlier is no longer working, and probably one of the reasons for this change can also be Mycoplasma contamination. Mycoplasma may not kill your cultures completely. Okay, so they may be growing, the cells may be growing, but then they change the metabolism of the cells. So there will be altered expression of various enzymes and proteins. In fact, cell cycle will be altered. Some cells may die also, but then yeah, so whenever you see change in experiments between when what you have already established few months ago to what is currently happening, then one has to be very careful, and one has to suspect whether Mycoplasmas may be a cause for this. Okay, so one has to definitely look for these things. So in addition to the FBS, which is the second major source of Mycoplasma contamination, and our own mouth and saliva, which is the primary cause of Mycoplasma contamination, another factor with which one can get this when we procure cells from other labs, or even from some vendors. So depending on how strict the people are maintaining cells there, one cannot always trust that what you are receiving from other labs is, especially from outside the institute, from a different culture unit, whether they are completely free from Mycoplasma or not. So in fact, just like how we quarantine animals when they are coming from a different source, in the same way cell cultures should also undergo quarantine when you are getting it from a different institute. And then one has to do a Mycoplasma testing before you introduce the cells to the rest of the incubator where your other cells are growing, because once Mycoplasma spreads, is introduced, it contaminates the entire incubator. Every person's cells will undergo complete damages. Okay, so one has to quarantine cells, test for Mycoplasma. If they are positive, then bleach it and eliminate it. No feeling guilty about it. And if it is negative, then one can put it in the regular cell culture, and one can be happy about that the cells are fine. And one has to always remember that once every few months, maybe once twice every year, one has to do check whether there is any Mycoplasma contamination or not. Of course, one thing I have told you, your results may be changing or not reproducible, then that itself is an indication. But whether there is any indication or not, one has to every year, twice a year, one has to check whether there is any Mycoplasma contamination or not. There are various methods to find whether there is Mycoplasma contamination or not, because once it is detected, sometimes we know of labs where they have discarded their entire cultures. Everything that is there has been completely discarded into the dump and then has been sterilized. In fact, people have frozen vials also, and therefore they have to completely discard the entire liquid nitrogen stocks also, because there is no way, if once it has entered, then there is no way to get rid of it. The only way to get rid of Mycoplasma is to basically bleach and throw that culture away. So there are ways to use some quinolones like ciprofloxacin and nalodixin and other things to get rid of these Mycoplasma, but then those are also topoisomerase inhibitors. They also cause inhibition of your cell growth, of your toxicity to your cells also. So it's very difficult to get rid of Mycoplasma, and the easiest is to basically throw the cultures only. So and if you have frozen vials also, they have to be thrown away. Okay. Detection is easier. There are various methods to detect Mycoplasmas. At least 2 different methods have to be one has to use. Okay. The classical method is the microbiological colony assays, where you make an agar plate just like your bacterial plates. You make it, and then you put the from your cultures, you put your from your media, you take it and culture that on it. Okay, if there is mycoplasma, and you have to wait, as I told you, because you have a slow long lag phase, you have to wait at least 1 week to 10 days before you interpret any data. Okay, so after 10 days, or at least after 7 days, you can check if there is any positive mycoplasma growth. Then you can see there are microscopic colonies, some 100 to 400 micron diameter size colonies. You can see they show what is typically called as a fried egg appearance. So if you have such kind of appearance underneath this on the surface of the agar, then that indicates mycoplasma contamination. Okay. A quicker method is by using real-time PCR, or where you are using it against 16S rRNA primers designed for it, for the Mycoplasma 16S rRNA. Okay, and one can also do hex staining. Okay, so this is another typical example or method by which one can detect these colonies. Okay, so I'm going to enclose or send in the Google Classroom paper from Texler, which is very good, and a Nature Protocols one, which I have indicated in the previous slide. So these are very good and very important papers that suggest both of you to read, all the students to read. Okay, and be aware that mycoplasma is a potential contaminant in the cell cultures, and one has to be very aware and cautious when doing cell cultures. Okay.